Hello everyone. I am honored to share with you our article published in iMeta, entitled Bavachalcone targets the transferrin receptor and sensitizes gemcitabine to influence bladder cancer progression. Urothelial carcinoma is one of the most common malignancies of the urinary tract. Bladder cancer can be classified, based on tumor invasion into the muscularis propria, as non-muscle invasive bladder cancer and muscle-invasive bladder cancer. For non-muscle invasive bladder cancer patients, postoperative intravesicolation is an important therapeutic intervention. Commonly used agents include Bacillus calmette-guerin, gemcitabine, mitomycin C, and epirubicin. In this study, using high-throughput drug screening and patient-derived organoid models, we demonstrated that bavachalcone effectively sensitizes bladder cancer to gemcitabine. Bavachalcone directly targets TFRC and EGFR, inhibiting iron influx and stabilizing TFRC on the cell membrane. By suppressing the iron-dependent ATR CHEK1E2F1 signaling pathway, bavachalcone downregulates the expression of the key gemcitabine resistance protein RRM1. In both PDX and orthotopic bladder cancer models, the combination of bavachalcone and gemcitabine potently inhibited malignant progression. We first constructed 4 bladder cancer organoid models and used bladder cancer cells and organoid models to conduct quantitative drug screening of the traditional Chinese medicine monomer library. We found that bavachalcone can sensitize cells to gemcitabine. Through in vitro studies, we demonstrated that bavachalcone suppresses bladder cancer cell proliferation and metastasis and induces DNA damage. In vivo experiments showed that bavachalcone combined with gemcitabine effectively inhibits malignant progression of bladder cancer. Using CMBR-activated Cepheros 4b, we found that bavachalcone targets TFRC and EGFR, and we validated these interactions using DARTs and CETSA. Using molecular docking and experimental validation, we confirmed that bavachalcone binds to the 416 to 460 region of TFRC. RNA-seq results showed that bavachalcone treatment inhibited pathways related to DNA repair, DNA replication, and metal ion transport in bladder cancer. Studies have shown that bavachalcone treatment reduces intracellular levels of total iron and ferrous ions. Bavachalcone can inhibit gemcitabine-induced DNA damage repair and the iron-dependent ATR CHEK1 signaling pathway. Patient-derived xenograft (PDX) models confirmed that the combination of bavachalcone and gemcitabine effectively suppresses malignant progression of bladder cancer. The study also found that TFRC and RRM1 are highly expressed in tumor tissues and are associated with poor prognosis. Transferrin binds extracellular Fe3 to form a TF-Fe3 complex, which then engages the transferrin receptor on the cell surface and is internalized via endocytosis. The released Fe2 enters mitochondria, where it combines with sulfur to assemble iron-sulfur clusters that support electron chain activity and ADP production. A portion of Fe2 also translocates to the nucleus, participating in DNA replication and repair, thereby activating the ATR CHEK1 signaling pathway and promoting RRM1 transcription. Bavachalcone blocks TFRC binding, preventing TFRC endocytosis and causing its retention on the plasma membrane, which inhibits iron influx. This reduction in intracellular iron impairs mitochondrial respiratory chain function and ADP synthesis, leading to attenuation of ATR CHEK1 signaling and suppression of RRM1 transcription. In summary, this study demonstrates that bavachalcone has the ability to enhance the sensitization of gemcitabine. Bavachalcone combined with gemcitabine can effectively inhibit the malignant progression of bladder cancer. Bavachalcone inhibits the influx of iron ions by targeting TFRC and EGFR. Bavachalcone inhibits the transcription of RRM1 by suppressing the iron-dependent ATR CHEK1 signaling pathway.